Hello friends, good evening. Welcome to Sam Biology class. Friends, today we shall discuss about Florigen. Florigen is a systemic signal that initiates flowering in plants. It was first described by Soviet Armenian plant physiologist Mikhail Chailakian, who in 1937 demonstrated that floral induction can be transmitted through a graft from an induced plant to one that has not been induced to flower. Later, Anton Lang showed that several long-day plants and biennials grown under non-inducing conditions could be made to flower by treatment with gibberellin. This led to the suggestion that Florigen is composed of two classes of flowering hormones such as gibberellin and anthycin. Gibberellin was considered as the limiting factor for long-day plants. It is synthesized when days are long. Similarly, anthycin was considered as the limiting factor for short-day plants. It is synthesized when days are short. To know the physical and chemical nature of Florigen, a number of known chemical compounds such as DNA, gibberellic acid, cyclic AMP and prostaglandin have been tested on vegetative plant to understand their florigenic activity. But none of these compounds exhibit universal florigenic activity. Because of this, initially Florigen was considered as the hypothetical flowering hormone. But more recent findings suggest that Florigen does exist. It is a systemic signal that initiates flowering in plants. It is a globular protein that is expressed in phloem of the leaf. After its synthesis in phloem of the leaf, it is translocated to the shoot apical meristem where it initiates flowering. In Arabidopsis, Florigen is encoded by flowering locus T or FT gene. Its homologs have been found in a wide variety of plants. All of them play a similarly important role. In rice, Florigen is encoded by HT3A gene. In poplar by PTFT1. In citrus by CIFT, and in tomato by SFT gene. As already described, Florigen is a globular protein. After synthesis in leaf phloem, Florigen is then translocated to the shoot apical meristem where it initiates flowering. This figure shows various interactions in the cells of shoot apical meristem that result in the formation of Florigen activation complex. In the cytoplasm of cells of shoot apical meristem, Florigen interact with 14-3-3 protein to form a complex. 14-3-3 protein act as Florigen receptor. Then the complex of HD3A protein and 14-3-3 move into the nucleus where it interact with a transcription factor OSFD1 to form the Florigen activation complex. The Florigen activation complex of HD3A, 14-3-3 and the transcription factor then interact with OSMADS15, a rice A petala 1 ortholog. This interaction initiates flowering. So friends, this is all about Florigen. For more updates, please subscribe my YouTube channel, Sam Biology Class, and also please share your comments. Thanks for watching.